Okay, so we are continuing the review for Bio 211 test number 3, and I'm picking up on a series of questions here. So we're just about halfway through the review, and I'd like to finish this thing up by first beginning to ask this one question. So first of all, remembering that this is Bio 211, it's Norwalk Community College, and this is a review for the third exam. But let's start with what are cranial nerves, and cranial nerves are of course chapter 14, and what is the function of each of them. Remember that I had you focus on where the nuclei of cranial nerves would be found. So that's the first thing you want to think about. And then the second is, what are their general purposes? If you took a look at the list, you'd notice that most of them are associated with special senses, right? But the remainder have to do with the interoreceptors we spoke about, say, in chapter 15. So when you're looking at how to answer this question, what are the cranial nerves and what is the function of each of them, there's a table inside your textbook that will give you the summary of which is sensory, which is motor, which is both, which is autonomic. And you want to pay attention to that, I think, probably more than anything else. Okay, because that's the distinguishing characteristic. I suppose this is the time where I point out yet again that the olfactory cranial nerve does not send sensory input to the thalamus. Remember, it goes directly or bypasses the thalamus altogether. But the second part to this question here, why do we tell you that the 9th and the 10th cranial nerves are the most important? And I think when you're looking at the actual table, or hopefully what you perceive when you're looking at the table, is that they are sensors for materials that we probably haven't thought about in a while, right? So hydrogen protons, carbon dioxide, okay? And what I mean by that is they're detecting how much of it is inside your bloodstream. And remember, the motor outputs now for these cranial nerves are to change, if you remember chapter 15, are to change the physiology of each of these. So if you think about what it means to have maybe too much carbon dioxide inside your bloodstream, what would you expect to happen to heart rate as a result of that? And this is why it's important to think about, cranial nerves 9 and 10. Okay? The next part to our question here in our review is actually multi-pieced. So I'm going to move this down here. So I've got some room to draw for you, or actually at least let's go like that. See if we can create some space here. Draw the brain and place the limbic system, diencephalon, RAS, and medulla oblongata in their correct locations, right? So you could think about this a couple different ways. Last night this wasn't working so well, so let's see how we're doing today. Still seems to be having some issues, but here's a brain. Think about how quickly it takes us to draw the brain. It doesn't take any time at all, all right? Yay. Boom, boom. Spinal cord, fantastic, right? But now we're asking, well, where's the limbic system, right? So where would you locate that in a midsection, or if you're going through it? Where's the diencephalon? Well, the diencephalon, at this point in time in the semester, there should be no mistakes with this, but if there is, that's okay too. We just got to fix them. So epithalamus, or pineal gland, is superior and posterior to the thalamus. And the hypothalamus is going to be anterior and inferior. I put a little box through that for us, right? The next part I want you to identify here is the reticular activating system. It's not just, where is this? I mean, obviously when you're reading these, you're thinking to yourself, okay, what is the limbic system? Emotional responses to sensory input, both somatic and autonomic. Diencephalon. What is the central sort of processing area of the brain? And then how do we regulate, say, endocrine responsibilities from nervous system inputs? Reticular activating system. This is a series of structures that help us figure out literally when something is worth paying attention to. And, you draw that through the midbrain. Yay. Boom. Like that. Let's put RAS there. RAS. Okay, and then the medulla oblongata. Well, here's our pons, here's our medulla, and of course I want you to pay attention to the fact that cranial nerves 8 through 12 are going to be found here, along with, say, the pyramids. Okay, so yeah, sure, you got cell bodies inside there, you have the pyramids as well, pyramids, pi. Okay, what's the significance of this? Okay, it looks like, should be, the central routing area for all sensory and motor pathways, ascending and descending through the spinal cord. Okay, that should be, I guess, obvious at this point in time. Now explain. I'm going to switch colors here. I'm going to move to green, and I'm going to draw in. Let's see what we can do here. Green. What sort of green do we want? I'm just going to go with some weird-ass green and see what we get. Okay, where do we actually make cerebral spinal fluid? Well, remember, inside the cerebral spinal fluid, we make the vast majority of cerebral spinal fluid inside ventricles. All right? And those ventricles have within them these structures that are called choroid plexuses that use ependymal cells. Whoops. Oh no, gosh, I hate when that happens. Okay. I'm sorry, we're going to lose this whole thing. So I'm just going to put ependymal cell here. Epend. Ependymal cells, it's supposed to be an E. And remember, those ependymal cells filter materials so it becomes cerebral spinal fluid. F. Okay. Now when I do this, it's going to erase the whole thing, so I apologize. Okay. Now, finally, so once you actually have it, remember, it's the cilia. It's the cilia that actually allow. Excuse me. Sorry about that. How it travels through the central nervous system. So in here, I will simply type in cilia on ependymal cells. Epend. Okay. Ependymal cells. Okay. The last part that's going to require us to draw again, use specific colors, okay, and add on to our sort of remind us the significance of sensory inputs, okay? Finally, explain how somatic sensory input travels into the brain and then sends somatic muscle response out of the brain to the effector system. So let's do this. Let's move away from green. Let's go back to neutral, sort of. Let's go. There we go. And we're going to draw a brain, and we're going to do this, and we're going to go do like that, we're going to go like that. And we're going to have the entire spinal cord. Yay. Put the pituitary gland in there. Cool. And with the basic structure of the brain in place here, remember over here is our cerebellum. Remember we have our arbor vitae, which is the axons of those neurons, and you actually have the gray folia. Okay, remember that the cortex of the brain here is also where we interpret signals that are going to ultimately end up at the cortex. And what I want to do is I want to create a scenario, artificial but important, okay, in that we need to, oops, somehow I managed to now do that. Where did my color go? There it is. Okay, let's draw that again. All right, now I need to change colors because even though it's artificial, I need you to understand that there is a first order neuron that has to be activated that will travel here. Remember, this is a myelinated unipolar neuron that will activate a second order neuron that tends to travel posteriorly, decuss somewhere inside there maybe, travel to the thalamus, where now we will activate the portion of the brain in that post-central gyrus for all sensory input to travel in. Okay, so somatic sensory input travels in. Check. Now we change colors because we're going to have to activate red here. We're going to have to activate the somatic motor cortex, which is then going to send the upper motor neuron around the thalamus, exit down through those pyramids if it's pyramidal, or around them if it's extrapyramidal, activate our lower motor neuron, which then travels through the anterior rootlets to now activate whatever the skeletal muscle tissue is that has to be moved. Okay? And chapter 16, is how do we look at each of the different types of, let's change color back to black here. What are the different types of, oops, I'm supposed to change back. Modalities. Okay, modals. Modalities, and that is pain, pressure, tickle, itch. All the characteristics of types of sensation that are traveling on unique pathways. Okay, So in terms of like orders of importance, first order, second order, third order, upper motor neuron, lower motor neuron. That's the most important stuff, right? You can't walk out of this classroom without knowing that. It's what's going to hold you in good stead for the rest of your careers. On top of that, knowing the types of sensory pathways. Where is it? Where's the delineation? What types of sensations are collected in the lower part of the body versus the upper part of the body? And if you have a face, what's specific to, say, facial integration of sensation? Like, why is the trigeminal nerve so important here? Okay, so when you're studying for the exam, try and make sure you understand the foundational information first, then layer on the additional pieces. Okay, well, let's get out of that. Let's put that over there. Let's open up the next slide. What do we have? What are the components to a reflex arc, and where will you find each of the components? Right, so pick up the pen again. I'm going to draw first the spinal column. Yay. And you should obviously remember the different pieces of the spinal column. And notice I'm just sort of being rather reckless here. Lateral gray horn, anterior, lateral gray horn, posterior, central canal, posterior. Just put a P there. I am getting upset with this. And this is anterior, right? And again, what I'm going to do is I'm going to first draw in. The structure is going to tell you exactly where you are. Here's that dorsal root ganglia. Okay, and here are our anterior rootlets. Oh, I don't know why this isn't working so well. But... Okay, and remember, it's a sensory reflex arc, so that means we have slightly different rules here than the previous explanation, where we're going to have some sensory input traveling in to activate. Let's change to green. Let's go down here and find a different looking green. I'm not quite sure that's green. I'm not quite sure what you guys see when I'm doing this, so there we go, green. Here's a cell body of an interneuron, and it's going to travel across here. And I'm going to now switch to red, because you know by this point in time, red means somatic motor output. So here's the cell body, and we're now going to exit out through the anterior rootlet, okay? Where we'll have our effector activated, right? And this means we can now think about... Let's go over here, put you back to that right there, so in case I come back to using you. But let's follow this through this way. Remember, sensory input travels through our sensory or afferent neuron, which will travel in. This is a unipolar neuron, which means the cell bodies are inside the dorsal root ganglia. We're going to now activate the cell body of an interneuron in the gray matter of the spinal column to then activate the cell body of an somatic motor neuron, which will then activate the effector tissue. Okay. Another thing we could do here while we're talking about this, we got a couple seconds, is remember that all of this is covered with connective tissue, pia matter directly attached. Then you actually have cerebral spinal fluid. Then you have the arachnoid matter and the dura matter sitting on top of that. Okay. Now there are many different types of reflexes. We did, what was it, 3 simple ones. We had 1 complex one. And by complex, I mean that you actually had to move one leg away from the stimulus of the tack, and you had to use the opposite leg on the opposite side of the body, which means that would be a contralateral response. And that's what we would see here. Okay, if I wanted an ipsilateral response, I'd have the interneuron activating a motor output on the same side of the body. So know the difference between ipsilateral and contralateral. Cool. Let's go over here. Now let's first of all do this. Get rid of all of that. What do we got here? Let's just answer these as if they were questions on an exam. What are the different modalities for stimulus for the nervous system? Again, we could just simply list those for you, and maybe that's what we should probably do. Make sure you understand what pain is. What type of receptor would that use? How about tickle? we didn't speak about what actually tickling is or itching versus. So one is physical stimulation versus, say, an itch, TCH, which could be chemical or physical. Chemical. All right. When you think about when a mosquito releases materials into your skin, it causes an itch, and that is histamine causing that response, right? And then there's just flat out, what else do we actually have? There's pain, tickle, itch, and temperature, TEMP, right? Versus, say, special senses. And we haven't actually covered chapter 17 yet, which means in lab, when we looked at the eyes and the nose, right? Remember, you actually collected light, L-I-G-H-T, which is then converted into electrical information. Okay, and that electrical information, information, okay, is what's going to travel on the neurons to those parts of the brain. Now, how does this actually fit in with the next piece to the question here, which is what is stimulation-induced adaptation? Remember the example I used in class is like, say, a perfume, parfume, right? F-U-M-E. And when you actually have an excess of it applied, you may not smell it, but when you walk into the elevator where everybody else is now moving away from you, it's because you have down-regulated the receptors. Down-regulation, D-O-W, regulation of receptors, while the new individuals smelling it for the first time haven't done that, so they will then down-regulate their receptors. Okay? It probably helps if I could reset. A-P-T-O-R-S, regulation. A-T-I, put a thing around that. Cool. I do think this is it, right? This is the last one. And again, we're coming to the end here. And obviously, this is the stuff that I've seen students have the most difficulty with. This by no means suggests these two 20-minute slide presentations for you are the only things you should be studying. This is the bulk of the material, though, that I've seen students not do well on. So let's use this last one. What are the different types of receptors located in your body and how are they classified? So you could literally draw an entire human body here. And I think I've done this for you in class, right? Where I did something like this. And I put your neck there. Arms. Correct anatomical position, so arms down. Torso. Legs. Not a perfect human being, but then again, I don't want to be Pablo Picasso. Okay, and then what am I actually asking here? Where are the different receptors located on your body? Well, remember, what I'm actually asking you about is what is a dermatome? So here you have a dermatome for your face, the trigeminal thalamic portion, and then you actually have that banding pattern that we looked at inside your textbook where you have specific receptors that are overlapping at the edges, meaning boundaries of receptors that are detecting sensation, okay? And what are the different pathways, sensory pathways that go to the thalamus? And this is a continuation of the original question, right? So let's change this question, this sort of open-ended essay-type question I had earlier for you, where a bug lands on your arm or something like that. You now have a first-order neuron traveling in. It then ascends to the thalamus, and then it will ascend to the cortex of the brain. Okay, maybe it goes to the other side. Makes no difference, right? I guess it actually does. Imagine I'm erasing this and say, look, it is going to be processed on the opposite side of the brain. Okay, good teachable moment there. Okay, that's why I stress thalamus here. Where did the decussation occur? And there's a specific type of pathway for that. And then, of course, what are the different motor pathways? So I'm going to change color here. I'm going to go to red. And now I'm going to say, okay, well, if we had a motor output, it would go around the thalamus. It would then descend where we'd activate the lower motor neuron. And imagine you're going to use this arm over here to swat the fly. So somehow we actually have to get the signal over here to now actually get the arm to move to swat the fly. So let me redo that whole thing so it's correct from the very beginning. Yeah, a minute or so. Okay, here's your body again. I'm just going to make this a lot easier. I think what happens is when you lift the pen up, it takes more processing time. So, it's exactly what it turns out to be. Look at that. Learn something new. What color did you give me? You gave me gray. I asked for blue. Okay, cool. Bug lands on arm. Whee! Okay. Sensory neuron detects that. That is a first order A-type fiber that travels to the spinal column. It will ascend on a specific type of pathway. It will go to the thalamus, and it will go to the opposite cortex of where the sensation is. Okay, your brain says, what the heck just happened to me? And it says, well, I want to get my other arm now to go swat that fly. So I'm going to start on the opposite side of the cortex, travel down around the thalamus, descend through the pyramids, then travel to the, now activating my lower motor neuron, activating the muscle groups that are required for both the upper limb and lower limb, and now you're going to swing that arm across and kill that bug. You should explain how that happens. Okay, if you have